This is our work that we've been doing for many years now, looking at a dual targeted CAR-T. So instead of just targeting the CD19 antigen, this CAR-T can target both CD19 and CD20. And the idea of this is through dual targeting, you can lead to a deeper remission and perhaps prevent relapse, which can occur sometimes due to CD19 downregulation if you target only one antigen. We have two abstracts here that are both as poster presentations, and this is sort of our developmenting this project over the last couple of years. So the first one was actually trying to optimize the manufacturing process. So we believe that how you manufacture the CAR-T cells plays a large role in how the CAR-T cells work from a functional standpoint. So what we wanted to look at was the optimal length of manufacturing. So we have an abstract titled 8 versus 12-day manufacturing process of LV20.19 CAR-T cells in B-cell non-Hodgkin lymphoma. In this trial, we enroll patients with diffuse large B-cell lymphoma, follicular, and marginal zone, and they were sort of sequentially enrolled on one of two arms. One was a flexible manufacturing process where if the cells were ready at 8 days after we started the culture process, we would stop manufacturing. And if they needed more time, we could go to 12 days, and the other was a fixed 12-day process. Our basic question here is, if we shorten the manufacturing process, does that impact the CAR from a functionality standpoint, from a safety standpoint, and also looking at it immunophenotypically? Our key finding was that we were able to achieve dose in all of the patients who were assigned the 8-day arm within 8 days. And immunophenotypically, we found that the CAR-T cell products that were manufactured in a shorter period of time, 8 days versus a 12-day cohort, that they actually had more stem cell memory type T cells, more naive T cells, versus those that sort of cultured until 12 days had more effector or terminally differentiated T cells. Well, why is this important? There's a thought out there that younger, more naive T cells can be more active, are better at killing, and can be more persistent in the body. In terms of response rates, it's hard to look at to really capture a difference because this was a very small sample size. We saw a slightly higher overall response rate, slightly higher CRS rates with the 8-day process, but again, difficult to take any meaningful data out of that. But what we sort of took away from this is that 8 days is probably the more optimal time to manufacture, and so we are now using that platform for future studies. And then we had a second abstract with that in mantle cell lymphoma, and within that abstract, we actually took that 8-day process and applied it to one disease. And so this was a phase 1-2 study in mantle cell lymphoma where patients were sort of manufactured using that 8-12 flexible process, and it looked specifically at this one disease subtype. And we enrolled 10 patients with relapsed refractory mantle cell. The median lines of therapy were 4, so suggesting that many of these patients were refractory. There are some patients who had prior allogeneic transplant, prior autologous transplant. All of them had been exposed to BTK inhibitors, as we talked about earlier, covalent BTK inhibitors. And some were actually exposed to pirtobrutinib because we had that clinical trial available at our center. So some had prior non-covalent exposure as well. Despite this, the bispecific CAR-T was very effective. The overall response rate was 100%, and none of our patients have relapsed with a median follow-up of 20 months. So I think this is very exciting data. We have some durability, a small number of patients, but the idea being that perhaps dual targeted CARs could be effective in diseases like mantle cell lymphoma.